Hello, everyone who's on and listening. David, are you Roxanne? Hello.
And he just joined.
Okay, great. Thank you, Sherry. Hi, Chris. There you are, David.
Hello.
Hello, hello. Welcome. We'll give people a minute. Some people are probably coming in just to hear you, David. So I don't want to start too early.
Sure.
But welcome to those of you who are just joining. We had a good time hearing lots of interesting updates and breakout rooms, and are very grateful to those of you who can join us on this July Sunday afternoon.
Yeah, we just had a big thunderstorm ran through here and my power went out for 30 minutes. It came on about 10 minutes ago. So
Oh my goodness.
I emailed Sherry and said, keep your fingers crossed.
Yeah. Oh, well, thank you.
I think we'll be okay, but if it had been a half hour later, you would have waited. Well, where is he?
Where is he?
Yeah.
Well, we're just starting here in Western Mass or mid Mass, and we're supposed to have floods by tomorrow night. There's no place for the water to go.
Well, we live on a hill, so we get flash flood warnings, but there's no danger, nor of any tornadoes, but rain, rain, yeah.
Yes, rain, rain, rain up and down the East Coast.
Hello, David. I'm Shippen. I'm his husband. And you taught me in last year's CPTR course, so thank you.
Okay.
You're welcome.
And you're color coordinated with the background.
I try to be color coordinated, but like a chameleon.
There's no
Okay.
white patterns in the background though, like there is on the shirt.
Yeah.
There it is.
There's our thunder. You sent it up to us, David.
No, you're too far north.
Okay.
David, where are you physically located now?
I'm Chapel Hill.
Oh, that's great. That's great.
Yeah.
So, should we officially get started?
Sure.
Welcome to David. Welcome to everyone. I'm going to read the bio that David sent us, just to give us a context. I personally am very excited to have David here. Couldn't have done the work that I've done through MAPS, and I know the work at Hopkins and NYU and lots of other places would definitely have not happened without David. So thank you so much for being here.
I was an evil chemist.
Yeah, right. Behind all this work. So Dr. David Nichols is currently an adjunct professor of chemical biology and medicinal chemistry at the Eshelman School of Pharmacy at UNC Chapel Hill. He's also the Distinguished Professor Emeritus at Purdue University School of Pharmacy and was the Robert C. and Charlotte P. Anderson Distinguished Chair of Pharmacology at Purdue. Beginning with his graduate studies in 1969, Dr. Nichols was among only a handful of scientists in the world who pursued the medicinal chemistry and pharmacology of hallucinogenic agents. In 1982, his laboratory carried out the first work elucidating the primary mechanism of action of MDMA as the release of a 5-HT from serotonin neuron terminals. In 1993, he was the founding president of the Hefter Research Institute, a 501c3 nonprofit that's raised more than $10 million from philanthropists to provide funding for early seminal studies of the use of psilocybin-assisted therapy for end-of-life patients because no institutional funding was available. His laboratory prepared the high-purity MDMA used by MAPS for its phase 1 and 2 clinical studies of MDMA-assisted psychotherapy for PTSD, the DMT used by Dr. Rick Strassman for his studies of the psychopharmacology of this substance, and the psilocybin for all the clinical studies at Johns Hopkins University involving psilocybin-assisted psychotherapy. And as an aside, I'm delighted to see that Bill Richards is on this call with us. So thank you, Bill, for being with us. Yeah, thanks. Since his retirement from Purdue in 2012, he's continued to publish in the field and has consulted for several biotechs attempting to develop psychedelics as therapeutic agents. Thank you, David. I'll let you take it from here. The talk title is Overview of Psychedelics from Prehistory to the Present. And Dave has been along on a lot of that, not prehistory, but certainly up
No.
to that.
Long
So,
enough, though.
great. Thank you so much. It's all yours.
Glad to be here. My talk is going to be, the first half is going to be just a background that much of which you will probably already know, but maybe not. So everybody's kind of on the same basis. And then I was asked to just basically field a lot of Q&A after that. So I've cut my talk to hopefully about 30 minutes, and then we have lots of time for questions if you have things you want to ask me. So let me share my screen. Make all this high technology work somehow.
Mhm.
Okay, so you heard the topic for the talk, and in the background, this is a picture that was painted by Alex Gray, who does a lot of very kind of psychedelic artwork. So I just thought that would be appropriate to start with. So this is a cover from last March's Chemical and Engineering News, which is the official organ of the American Chemical Society. And you can imagine that for most of the years I was in this field, we never saw anything about psychedelics in their magazine. So they've been accepted and broadly written about in lots of different venues. So as you know, it's been called a renaissance of psychedelic research. So there's a lot of interest now, a lot of clinical studies, a lot of preclinical. In fact, I can't even keep up with all the literature nowadays. LSD was actually scheduled in Nevada and California in 1966. And then with the passage of the Controlled Substances Act in 1970, which was enacted by the Nixon administration to suppress voting by hippies and Black folks, we saw a decrease in publications. This ends about 2020, but the publication list has increased. I have thousands now of downloaded publications that have come out with respect to studies of psychedelics, chemistry, pharmacology, clinical studies, etc. So there's been a rapid rise and increase, and the media has been covering this quite a bit. The thing I want to say, first of all, is that psychedelic agents, and the classical psychedelic agents at least, are interesting and different from other types of psychoactive compounds because they reliably induce or compel states of altered perception, thought, and feeling that are not or cannot be experienced otherwise except in dreams or at times of religious exaltation. There's actually some debate about what psychedelics are. There are a lot of people that want to include some atropine type compounds, salvinorin, ketamine. I'm going to talk just about the classic psychedelics, which are those that activate serotonin 2A receptors. And you'll recognize that this is a very unusual definition. This is a formal pharmacologic definition of Goodman and Gilman's 8th edition. It was also in the 7th edition, which is sort of the textbook for pharmacologists. So drugs that produce experiences like dreams or religious exaltation, obviously that's a very unusual definition. And these are very unusual class of drugs. They've been known throughout history and have also been adopted by shamans and witch doctors and so forth thousands of years ago. This actually, let's see, if we can get somewhere. This is about 2 weeks old. I checked clinicaltrials.gov a week or two ago when I was putting this together, and there's 143 clinical trials of psilocybin currently registered with the FDA, 136 for MDMA. These numbers are probably slightly different as of this today, but when I started my research in 1969, well, there wasn't a clinicaltrials.gov at that time at all, but had there been, there wouldn't be any clinical trials of psilocybin or for MDMA. So this is sort of the state right now of clinical research, looking for people to get approval from FDA for what they're doing with psilocybin, MDMA. Also, we could have DMT added, LSD, and some others, but basically, these are the major things that are being investigated today. Just looking at natural sources of these psychedelics, we have 3 chemotypes: phenethylamines, tryptamines, and lysergamides. The phenethylamines, this is a little baby peyote cactus. This is a source for a substance known as mescaline, which is a 3,4,5 trimethoxy. These are methoxy groups, And I may spend a little more time on definitions because I don't know how many people are really chemistry favorable. Usually my audiences, if they're physicians or pharmacologists, don't really know much chemistry. So I may spend a little more time than I would ordinarily. But this is mescaline, 3,4,5-trimethoxy phenylethylamine. And it's a classic psychedelic that was used. It occurs in that peyote cactus. Not very potent. The dose of this is perhaps 200 to 300 milligrams, is used by services of the Native American Church as a sacrament. They eat the peyote cactus. Then we have tryptamines. This is an example of psilocybe. There are about 200 genus or species of psilocybin containing mushrooms. This is a tryptamine, again, an aerial system, 2 carbons away from a basic nitrogen. Mescaline is an aerial system, 2 carbons away from basic nitrogen. This is a common template in drugs that have an action in the central nervous system because it resembles to some extent neurotransmitters. The phenethylamine is the structure of norepinephrine and epinephrine, and this tryptamine structure is the basic template for serotonin. So if we have nothing attached over here, this would be N,N-dimethyltryptamine or DMT, which I'll talk about briefly. If we have a methoxy, this is a methoxy over here. If we have one hooked at the 5 position, that would be 5-methoxy DMT, which has also been called toad, which is a very potent psychoactive compound. It's usually smoked. It's not orally active. DMT is not actually orally active either. It's usually smoked or insufflated through the nose. It's a synthetic molecule, but also occurs naturally. And it occurs in the venom of toads. And so you can take the parotid glands on the side of a toad's head and squeeze them, and they'll squish out this white milky toxin. Squirt it onto a piece of glass, it'll dry, and scrape it off. And it contains a bunch of toxic things, but they mostly are broken down if you hit them with a flame. So they put this toxin into a pipe and smoke it, and the 5-methoxy DMT is vaporized, and the other things are destroyed. If we have at the 4 position, if we have an oxygen up here attached to a phosphate group, that's psilocybin. Psilocybin is a prodrug for a drug known as psilocin, which has the 4-hydroxy. So at this 4 position, if you have an OH there, that's psilocin. And that's actually the active species if someone eats psilocybin. The phosphate on this oxygen of psilocybin is cleaved off. And then we have lysergamides. They originally came from ergot fungus. So this is an ergot-infested stalk of rye. These black things are called ergot, grains of ergot. And what they are is actually the inert in form of ergot. It's called the sclerotia. And so the ergot will produce the sclerotia to survive and sort of hibernate. If you peel one of these off and put it into a medium where it is encouraged to grow, little mushrooms or fruiting bodies will grow out of these. So it actually is a fungus, and it's very toxic. Bread during the Middle Ages often got, the grains got infected with ergot and produced outbreaks of ergotism called St. Anthony's fire, which if the bread was ingested in a significant amount would cause vasoconstriction of the fingers and toes, and they would turn dark and black and fall off and become gangrenous and be very painful with a burning sensation. So sometimes it was called St. Anthony's fire. And St. Anthony was a patron saint of people who had suffered from ergotism in the Middle Ages. Now, if you grind these ergot kernels down, you can extract ergot alkaloids out of them. And one of the ergot alkaloids we're interested in are the lysergamides. This is an ergot system. It's a 4-ring system that's aromatic here. Again, if you look, you see like tryptamines, you see the indole nucleus over here, 2 carbons away from a basic nitrogen. That's a common theme in CNS active compounds and in the psychedelics. There are lots of different ergot alkaloids. This actually is a derivative of lysergic acid. So lysergic acid would have a carboxy group up here, an acid group here. If these R groups are ethyl groups, 2 carbon ethyl groups, then this would be the diethylamide. This is an amide function, diethylamide of lysergic acid. It has a specific stereochemistry here designated as R, and it's plus. So this is and this is R here, 8R. So this would be a diethylamide of lysergic acid, or LSD if these were ethyl groups. And of course, you see they also have this tryptamine fragment within them. The Aztecs used, when they couldn't get magic mushrooms, they used Turbina corymbosa, which they called ololuki. And the same component that's in ololuki is in Hawaiian baby wood rose in the seeds, and it's ergine or lysergamide. It's lysergic acid, amide. You see, we don't have the two ethyl groups here. This compound is not as potent as LSD. LSD is a dose of maybe 50 to 100 micrograms would be typical. And ergine or lysergamide, you might take 2 or 3 milligrams, and it does not have qualitatively the same effect. But it was used when the mushrooms weren't available by the Aztecs. And actually, there are seeds. The production of organoalkaloids doesn't actually occur in the plant itself, but rather it's symbiotic with the seeds. So if you take morning glory seeds, or there are heavenly blue morning glories that were used back in the 60s, you grind up the seeds, you can actually extract out ergine, which is lysergamide, which is psychoactive. Now, I mentioned dimethyltryptamine. You've probably heard of ayahuasca, and I don't know if you can see this on the top of my screen, maybe not, but ayahuasca is up here. Ayahuasca is a mixture of two things: dimethyltryptamine, DMT, which is produced in the leaves of Psychotria viridis, also called chacruna, and it's not active orally. If you take DMT orally, it's broken down by monoamine oxidase in the liver, so it's not active. But there's a vine that also grows in the Amazon called Banisteriopsis caapi. And if you shred the bark, if you cut this down and shred the bark, that's what you see in this picture here, these red pieces. They're bark from Banisteriopsis. And they produce an alkaloid called beta-carbolines, alkaloids called beta-carbolines. And beta-carbolines will block the action of monoamine oxidase in the liver. So you put the Banisteriopsis caapi, the shredded bark, into a pot with some chacruna leaves. You add water and boil the dickens out of them, filter out the plant material, and you get this brown, bitter goo. There's extract called ayahuasca. And what it is actually, the presence of the beta-carbolines in this mixture renders the DMT orally active. So what you actually have in ayahuasca is a sort of orally active preparation that contains DMT. Now, where do the psychedelics act in the brain? They're very powerful. So we would expect that they might have a site of action that would be fairly important. They interact with G protein coupled receptors. Phenethylamines primarily activate serotonin 2A and 2C receptors. Tryptamines activate serotonin 2A, 2C receptors, and also 1A receptors. So 5-methoxy-DMT, for example, is a fairly potent agonist at serotonin 1A receptors, whereas most of the phenethylamines, like mescaline, primarily activate serotonin receptors. And LSD is a fairly promiscuous drug. It interacts and activates or blocks a number of other receptors. The thing they all have in common is the fact that they activate the type of G protein coupled receptor called the serotonin 5-HT2A receptor. And we've done lots of studies in vitro and animal models and in humans. If you give a drug which blocks or antagonizes the serotonin 2A receptor, you can prevent the effects of psychedelics. So we're pretty sure that this is the key receptor that's involved in the psychoactive effects of psychedelics. So where is that receptor located? These are PET imaging studies done from Copenhagen in Gitte Knudsen's lab. And so she's used positron emitting ligands that bind to the various receptors. And you can see the density here. So here's the SERT, which is the serotonin reuptake inhibitor. That's how SSRIs like Prozac work. They bind to that. The serotonin 1A receptor, 1B receptor, and the 4 receptor. And you can see of all the receptors in these examples, there is more serotonin 2A receptors in the brain than any other type. And you can even see the density at the bottom in picomoles per milliliter, 60 for the 2A versus 50 here at the 1A versus 9 or 30 or 3. So they're very highly expressed in these critical areas in the temporal lobe and the prefrontal cortex. And the cortex is where a lot of the action is thought to take place. And this is a view looking from the medial side. You can see they're inside and also back here. Also, the visual cortex in the back of the brain is right here, primary visual cortex. It also has a lot of serotonin 2A receptors. And that accounts for some of the visual effects that these psychedelics will produce. If we actually look into the brain at where they're expressed within the cortex, you see this is ketanserin, which is tritium labeled. And what they did was take a slice of human brain and incubate it with a solution of radioactive ketanserin. And then they rinse it off and they put a photographic plate on and develop it. And the density is expressed by color here. So this is high levels of expression where it's red. And what you can see is the outer layers of the cortex all the way through the prefrontal cortex, through parietal and back into the visual cortex. You have a high expression level of serotonin 2A receptors. And they're expressed on apical dendrites of cortical pyramidal cells. So within the cortex, the computational units are called pyramidal cells. And you can see them here. They have a kind of a triangular or pyramidal shape. And the apical dendrites are going into these pyramidal cells. And you can see from the density, these are antibody stained. There's a high expression density of serotonin 2A receptors on these apical dendrites of cortical pyramidal cells. And what happens when they're activated is these cells become more sensitive. They become depolarized more easily than they would normally. So what we have is an increased gain or amplification factor in these cells, so that levels of signal that normally would be fed into the cortex and would not be perceived during conscious awareness, by activating these pyramidal cells, they can pick up levels of signal that normally would be below the level of conscious perception. This is a mouse somatosensory cortex, and it shows you again, this is the outer layer of the cortex out here, layer 1. And then you would go, as you go interior, these are all inhibitory cells. We get down here to where these are the pyramidal cells. The apical dendrites are in layer 5 here, going into these pyramidal cells. And these red traces are all excitatory. So basically, if you think of the brain as like a parallel processing computer, these are all stacked together in parallel arrays throughout the cortex. And they're the major computational units. So you have a lot of, all your sensory information is sent into these cortical pyramidal cells. And somehow they put together our impression or our perception of reality by integrating all the inhibitory and excitatory inputs that are in this area, in the layer 5 of the cortex. Now, inside the brain cell, what happens? So this is a G protein coupled receptor. This is actually the serotonin 2B receptor, but the 2A receptor is almost identical. This is actually the receptor. And what I've done is I plotted out the alpha helices of these protein units. And they're also superimposed on that are the actual van der Waals radii. They're transparent, so it's a little bit messy. But you can see the alpha helices. These G protein coupled receptors are 7 of these alpha helices packed together in a bundle. And the width of the neuron membrane would be about approximately equal to from about here near the bottom to the top. So they're embedded in the neuronal membrane, and they have this architecture of these 7 transmembrane helices, and there's one helix inside, a helix 8, that runs parallel to the inner leaflet. Normally, they associate with a heterotrimeric GTP binding protein. So this is the inactive receptor. So we have heterotrimeric means 3 different heterotrimers, G proteins. So we have a G alpha subunit, a G beta, and G gamma. The G alpha subunit normally binds GDP, guanosine diphosphate. And when a psychedelic activates the receptor, there's a change in the way these bundle together. They change their overall shape or conformation, and that leads them to dissociate from the G alpha subunit. And it extrudes GDP and binds GTP and dissociates from G beta and G gamma. Beta gamma will then interact with signaling molecules, for example, potassium channels. The G alpha subunit then with the GTP bound will then go and interact with other types of enzymes, for example, phospholipases. And it will keep activating that until the GTP inside here is broken down. So this G alpha subunit has an intrinsic GTPase. That means that once GTP is bound, it acts like an enzyme to start breaking down the GTP. When all the GTP is broken down, this G-alpha subunit no longer is effective in causing a signal, and it will go back and reassociate with the receptor and the beta gamma subunits. So that's sort of the mechanics of it. Now, I'm not going to spend any time on this, but lots of things happen when you activate the agonist. Here's the G-alpha beta gamma subunits I talked about, that they dissociate from the receptor. The beta gamma actually does certain things within signaling. The alpha subunit, here I'm showing it interacting with phospholipase C to produce an isosulfide triphosphate. And then you also get PKC activated. That can lead to activated ion channels, arrestin association, lots of other things, activated kinases, leading to activation of BDNF and TrkB. These are thought to be responsible for the antidepressant effects. There's also intracellular signaling, then leading to spinogenesis, the formation of new dendrites on receptors, as well as the release of calcium, intracellular calcium. So this is a real biochemical mess. We don't really know which of these are necessarily responsible for the psychedelic effects. And people are intensively looking at all these different signaling components to try to figure out what is it within the neuron that's activated by a serotonin agonist that leads to these changes, and which ones are responsible for the psychedelic behavioral effects. Now, I'll mention briefly that I founded the Hefter Research Institute in 1993, and we funded about $10 million worth of money from philanthropists since 1993 to fund some of the earliest clinical studies of psychedelics. Obviously, when I was a graduate student in 1969, nobody was doing clinical studies. Well, I take that back. There was one clinical study that was ongoing at Johns Hopkins. It was the last study that was carried on, and it was not terminated until 1976. But other than that one study, which was sort of the last one, nobody was doing clinical studies. So when I started the Hefter Research Institute, I got together a bunch of my scientific colleagues, and I said, nobody's doing clinical research. That's the really interesting stuff. Why aren't they doing it? Well, they weren't doing it because after the Controlled Substance Act was passed, you couldn't get money as an academic to fund clinical studies. They were generally viewed as dangerous substances. Nobody would study them. As an academic, you were basically told, if you want to work on psychedelics, it's going to be the death of your career because you won't get funding for your research. I was lucky to get funded by NIDA, National Institute on Drug Abuse. And I had funding from NIDA for 28 years, but mostly to study the pharmacology and medicinal chemistry, basically asking questions like, what is the receptor? How do these drugs interact with the receptor, and what happens? And can we develop other models? We did a lot of structure activity studies, looking at modified molecules, taking mescaline and making changes, taking tryptamines and making structural changes, taking lysergic acid and making various derivatives of lysergic acid to try to understand. And then we did mutagenesis studies later on with the receptors to try to find out what it was about these ligands that was activating these receptors. So the clinical studies, these are some of the people that joined up. George Greer was a psychiatrist, Mark Geyer, a PhD, Roland Griffiths, who you probably all have heard of, Charlie Grobe, Dennis McKenna, Stephen Ross at NYU, Franz Wollenweiter in Zurich, and some others that supported us. And I just mentioned just a couple of studies. The first study we supported was Charlie Grobe's study at UCLA in advanced stage cancer subjects. Unfortunately, the dose he used was too low, and his paradigm wasn't optimal. So he didn't actually see significant effects until 1 to 6 months afterward, where he saw a reduction in anxiety. The two studies that were most well reported were these studies at Johns Hopkins led by Roland Griffiths, looking again at life-threatening cancer diagnosis with psilocybin-assisted therapy. N51, that was a good study. It was double-blind, placebo-controlled, published in 2016. A similar study out of New York University led by Dr. Stephen Ross, published with 29 subjects in 2016. These were the two biggest studies that talked about psilocybin having effectiveness in these end-of-life studies. There was also a study out of Imperial College that was published in 2016 by Robin Card Harris. It was a smaller study with, again, an N of 12, and he saw fairly dramatic results. We also supported studies of looking at psilocybin and smoking addiction. That study's been expanded now by Matt Johnson. Alcohol use disorder by Michael Bogenschutz, who was then at New Mexico, who went to New York University, has now published a big study with 96 and shown significant anti-addictive effects. And some studies of cocaine addiction, which haven't been published yet, out of Alabama. And then general studies by my son at LSU, looking now more recently at anti-inflammatory effects. And then Franz Vollenweider, who published the most papers involving clinical studies of psilocybin at the Zurich at the University of Zurich. He actually had a Hefter Zurich Research Center set up there, and then some others. But basically, that's what I want to cover science-wise. And just to mention that this is a cartoon that I thought was humorous, and it shows people going to get psilocybin for their CNS effects, depression, anxiety, rather than the standard pharmaceuticals. So with that, I'll thank, I had a number of graduate students and postdocs over the years. Again, I was funded for 29 years by NIDA, 28 years by National Institute of Mental Health to look at dopamine agonists. And then I also did early work on, I had about 12 years of study for the early work on MDMA, the preclinical work looking at the mechanism of action, making analogs. I also named MDMA as a new class of psychedelic or psychotherapeutic agent called entactogens. So if you see the word entactogen, that's a word that I cooked up to separate the MDMA, which at the time it first came out was categorized by the DEA as just another psychedelic amphetamine. And I knew it wasn't, so I spent a lot of time to prove that it wasn't. It was a novel class of psychoactive agent. So I think that probably is my last slide. So I'll be happy to answer questions as well as I can. So hopefully that was something that you were interested in, found interesting, and have some questions. And I was told to stop to allow Q&A. So there you go.
Great.
Great.
Thank you, David. I'm sure questions will start coming in. I didn't ask people to put them in the chat, but if you can do that, and I'll probably call on you to ask the question because it's much, unless you don't want me to do that. I have one question just from the last thing that you mentioned about the name entactogen. Can you give us some story about how you came up with that name and how that happened?
So when the DEA first, MDMA was used, it was called ecstasy, was used widely at raves, really made people feel very euphoric, in touch with other people. And the DEA, they had a whole program where they said, well, they had a guy from the DEA and he said, this is just another hallucinogenic amphetamine. They wanted to throw it in the trash bin of other hallucinogenic amphetamines. And by that time, I was convinced that it wasn't just another hallucinogenic amphetamine. I had a lot of familiarity with hallucinogenic amphetamines. In fact, I had developed the patented synthesis for making the isomers. So you have a right and a left hand for many chemical compounds. They're called isomers. So you have a D and an L, plus and minus. And it turns out that the psychedelic amphetamines, it's the minus isomer that's active, not the plus. With MDMA, it's the plus isomer. With amphetamine and methamphetamine, it's also the plus isomer. So we did a first study we published in 1982 where we showed that what MDMA did was release stored serotonin from neuronal endings. It didn't have this postsynaptic effect of serotonin 2A receptors. It was different. Its action was more like methamphetamine or amphetamine, but they're psychostimulants. They have their own category. So Ralph Metzner had come out and called them empathogens. And I kind of didn't like empathogen very well because it sounded like pathogen to begin with. And he said, well, they produce empathy. Well, they do produce empathy, but in a therapeutic context, you don't need the patient to have empathy for the therapist. You need the therapist to have empathy for the patient. So what do they do? And then I thought and I knew a lot about MDMA by then. I had George Greer had talked about it. I had a lot of stories from people, personal accounts, etc. It seemed to me like people were using it, at least in therapy, to go in and address painful emotional material inside, things that traumatic events and so forth. So they were reaching inside and getting access to that. So An and gen is inside and to produce, and tacto was taken from the Latin root for tactus, to touch. So I said, oh, they produce a touching within. That was the connotation of that. I still prefer that, and it's actually becoming, I hope, favored. I did a study, I did a search recently of a National Library of Medicine, compared entactogen with empathogen, and entactogen outscored empathogen by 4 or 5 to 1. So in scientific publications, and it didn't sound very scientific to me. I mean, this sounds kind of trite, but empathogen, but entactogen kind of, well, what's that mean? Well, it produces a touching within. So that's where the term came from. And I wrote an original article proposing that, and then for Archives of Psychiatry, last year I published another one, a review of how that came to be and why we called it that, et cetera. So I hope that, well, personally, I hope it takes off, but I think it's become the favorite term for these classifications. And you can use it in a scientific context without anybody going, empathogen, that sounds kind of goofy. So that was it. With all due respect to Ralph Metzner, who was a very prolific researcher and passed away recently, but I just didn't like empathogen.
Great. Thank you for that backstory. That's great. And Chris Germer has a question he would like to ask you.
Okay.
Yeah, so thank you so much. Oh my goodness, for your work. It's impossible to imagine this field without your work. And honestly, I had no idea what you've been up to. So what an honor to spend this time with you. And the title of that journal, chemistry journal, was about psychedelics without the trip. And I remember seeing a little study with mice who were sort of twitching, which meant they were tripping, and those that weren't twitching and so forth. And it really does seem to me that psychedelics without the trip would really have an enormous impact on the integration of psychedelics into psychopharmacology. I'm just wondering, what's the status of the research? What are your thoughts about that? I know Bill Richards is on this call, and you know, he has been very clear that a mystical trip is essential for positive outcome. Just love to hear your thoughts on that.
Yeah. So there's this popular meme that, at least in regulatory circles, that people don't want to experience this trip. It can alleviate depression, anxiety, etc., but people don't want to have the trip. If you talk to people that have actually taken a psychedelic and you say, well, would you have not wanted to have this experience? Most of them will say, oh no, it was one of the most valuable experiences of my life. And you know, in the study that Roland Griffiths published in 2006, which were normal people that were given psilocybin, I forget the percentage, but a huge percentage of them placed it among the most important and spiritually meaningful experiences of their lives. And I'm sure if you went to those people and said, well, what if we took that experience away from you and it still made you feel okay, feel good about yourself, but took that away? I think most of them, and I have some experience, I won't talk about that very much, but most people that have had a psychedelic experience that was very powerful and positive would not want to lose that experience. Now, I know some of the people that are really pushing this psychedelic without a psychedelic effect idea. I suppose, if you just want to compare taking an SSRI like Prozac, for example, or citalopram, for many people, if you could just give them a drug that worked like that and alleviate their depression, I think that would be completely fine. If people can do that, I don't know if they can. I'm not sure I agree with that. But for people who are addicted to alcohol, tobacco, or people especially who are at end of life and are facing the existential crisis of their imminent death, things happen with these psychedelics that don't happen with just a pill. So, and Bill Richards could probably address this easily, but give somebody who's facing the end of life and existential distress a psychedelic, and they get new perceptions about how important their life has been. They reconnect with, if they've got estranged family members, they reconnect with them and say, I miss you so much, I love you, I wish we had connected, etc. and they get a different perspective on death. This was discovered with Eric Cass back in the 60s, that people who got LSD who were dying had a new perspective on their death. They lost their fear of death. They had reduction in their need for pain medications. So there's something that happens that nobody really understands. They get a different perspective. Death does not become so frightening anymore. And I don't think you can take that away with a pill. And this is a whole experience they go through, and they re-examine their life. They do a life history and so forth. And the same thing is true for people that have been addicted to alcohol or tobacco. I heard a talk by a fellow who had been a pretty heavy alcoholic, was done in a New York University study. And he said, I constantly was obsessed with trying to find alcohol. If I went someplace, I would go out and scatter around and find out where are the bars I can go to when I need a drink. And his life was consumed by this need for alcohol. Well, he took psilocybin under a therapeutic regimen and looked at when he started, why he couldn't stop drinking, what prompted him to keep drinking, what was the reinforcement for him, how he damaged his relationship with his parents and his friends. A woman, the same thing, a woman who was in the first study that Michael Bogenschutz did, I saw an interview with her, and she said, I'd come home every day after work tired, and I'd immediately sit down and have a couple of drinks. And I would just ignore my kids. They wanted to eat dinner, here's mom's home, and then I'd be drinking and I'd be ignoring them. She said, under the effects of the psilocybin, I realized I was damaging my family. I was damaging my children. And she said, now I come home, I connect with my kids, I fix dinner, we sit, and her whole life had been changed. I don't think a pill will do those kinds of things. That's part of the whole process of what the psychedelic does, the behavioral effect. So for somebody who has uncomplicated depression that normally would be given an SSRI or an SNRI, something like that, if they can come up with a drug that does the same thing, whether or not it has, their idea is to try find things that activate the 2A receptor, which is what's thought to be mediating the antidepressant effects. If they can find it without producing a psychedelic effect, there's probably a huge market for it. A lot of people don't, they've heard all the horror stories about psychedelics or LSD or whatever, your brain melts or you jump out windows or whatever. They're afraid of it because of all the negative press and hysteria. So if they can do that for uncomplicated depression, endogenous depression, perhaps even for some kinds of anxiety, I think that's fine. But for many of these indications, for substance use disorders, for existential crisis, even for obsessive compulsive disorder, eating disorders is another. We don't know, we don't have the data, but there are eating disorder studies going on now. One of the first things that Hefter wanted to do was an eating disorder study, because we thought if you give people a psychedelic who have severe eating disorders and they're emaciated, you can put them in front of a mirror and say, look at yourself. What do you see? Oh my God, I'm dying. There would some kind of be a conscious perception of what they were doing to themselves. And we think the obsessive compulsive disorder, you get in these obsessive routines. I'm not sure that a pill will work for these things as well as having actually the psychedelic experience. So that's my take on that. If they can do it for uncomplicated endogenous depression and doesn't produce a trip, that's what SSRIs do, SNRIs do when they work. They're not very effective, so something more effective, I think that's fine. But I think in other cases, if I was dying in an existential distress, I wouldn't be looking for a pill. I'd say I want to have an experience. I want to feel what it's like to reconnect with, my concept of God, whatever it is. So that's a long-winded way of answering your question, but I hope I've done it.
Thank you. Can I just ask two very short follow-up questions?
Sure.
Yeah, one is I'm interested in the status of the research on psychedelics without the trip, and also the mechanism. So what I heard, for example, was neuroplasticity might be present even if there's not a trip, and then combining that with contemplative practice, in other words, like a meditation practice, might be something for people who are afraid of trips or who might not be otherwise indicated to take a full psychedelic journey. So status of the research and also neuroplasticity.
Yeah, it's been known for a long time. If you look at the brains of people who are depressed and then they've died, you look at the areas, the hippocampus especially, you see a reduction in the number of dendrites, those neuronal projections. And now in animal models, and they know from people that have been given an SSRI and recovered from their depression, you have resprouting of those neurons. So there are increased neuronal connections. So that's a neuroplasticity, meaning that they increase the sprouting of new neurons and dendrites. So there's more of a connection there. The problem really is a translational one. Most of those studies have been done in mice. And mice are not nearly as translational to humans as rats are, but mice are affordable. When I started my research, rats were maybe $2.50 a piece. We use lots of rats. We use drug discrimination as a model. Rats are now like $50 or $60, and mice are like $2.50 and $3. So in terms of a budget, and mice, this head twitch assay that people use in mice is a very inexpensive assay to run. You just, you set up a video camera and you look at their effects. No one is really sure whether the antidepressant effects in mice translate to humans. So this is a big problem, the translation problem. We know that in humans that respond to an SSRI, they have neuroplasticity, increased sprouting of neurons. So if you give psilocybin to a mouse, and then you examine the rat of the mouse, you also see increased dendritic growth, neuroplasticity. But we don't know, that's proposed that that's a mechanism for the improvement in the psychopathology. But we don't know for sure because mice are not very translational. There's a big problem now. NIH is having trouble finding useful models. If you think about, okay, how do you model depression in a mouse? So one model of depression is you hang a mouse up by his tail and he struggles for a certain length of time and then he stops. But if you give an antidepressant, he'll struggle for a longer period of time. Or if you put a rat into a tank of water, he'll struggle and then he'll give up at a certain point and just in despair. Or if you give an SSRI, he'll swim for longer. So these are the kind of models they have. And rats are more amenable to translational studies than mice are, but everybody's using mice. So there's this big question of, do these mouse studies really mean anything? But a lot of people, there are at least two or three biotech companies that are trying to find serotonin 2A agonists that don't produce the head twitch in mice, for example. But I have kind of a problem, a perspective that's a little different. If you think about where the serotonin 2A receptors are located on these apical dendrites of corticopyramidal cells, and if corticopyramidal cells are the computational units in the brain, what happens when you change their electrical properties? Can you do that without having a CNS effect, without having a behavioral effect? And I'm not sure you can, because those pyramidal cells, they are the most important computational units in the brain. They're, what in our prefrontal cortex, that's what puts everything together and provides our perception of our consensus reality. Now, can you change the electrical properties of those neurons? Can you make them more excitable without producing a behavioral effect? That's a basic issue I have with that whole idea. I'm not sure you can, but there's lots of research trying to find things that, and the original research that was done out of David Olson's lab, he made some compounds that he thought activated the serotonin 2A receptor. And so he said, but they didn't produce behavioral effects. He said, oh, we've got something now that activates the 2A receptor that doesn't produce behavioral effects. But in actual fact, the compounds that he first started his research program with, and actually started a company called Delix with, they don't actually activate serotonin 2A receptors. He didn't do the pharmacology right. Now that's all been corrected, so now he's kind of backtracked and all. But the original premise of that was that these were 5-HT2A agonists that didn't produce behavioral effects. But in fact, they weren't 5-HT2 agonists. And Brian Roth has the lab where I'm a guest now, where I work. And Brian had both of the compounds that were both first synthesized that David Olson claimed were, psychedelic, non-psychedelics. And they didn't activate the 2A receptor at all. But what they did do is they block the reuptake of serotonin in the serotonin terminals, which is just exactly what SSRIs do. So he may have been seeing an antidepressant-like effect as a result of the fact that they were blocking the serotonin reuptake here in the same way that SSRIs do. So it's a complex situation with lots of nuances. I'm not convinced they'll be able to do it, but more power to them if they can, because we need something to treat depression. And SSRIs are not very effective. A lifetime on them is not a very useful life. And people have a lot of side effects. You know, they lose their libido, their consciousness gets screwed up. So anyway, another long-winded answer to your question.
Wonderful, thank you.
Sure.
Greg Fournier, you have a question. Would you like to ask it?
Sure, thank you. So I've been so curious about why psilocybin is at the forefront of this renaissance, if you will, and substances like DMT are taking kind of a back seat with what it seems like right now. I mean, clinically, it would seem that DMT would make a lot more sense than the psilocybin with the short action, and it could be something that could be weaved into someone's life, you know, weekly sessions to do this, where psilocybin is it's a big, much larger commitment, you know, for people to undergo, and it's kind of prohibitive to do it on a regular basis, I guess is one of the questions.
Yeah, you've hit on a big problem, which is scalability. If you think about the studies that have been done with psilocybin, it's a 4 to 6 hour duration of effect, which means you bring somebody in the morning, they're there all day long. If you get 2 people there as sitters, a clinician, a doctor, whatever. The first studies that we did, I asked Steve Ross at New York University, I said, what's the estimate for the cost per patient in your studies? And he said, it's about $25,000 per patient.
Wow.
I'm not going to get insurance companies to pay for that. So monetizing that and a level that's affordable to just the average guy on the street that would, you know, normally be depressed and get SSRI, being able to make it affordable is a big problem. We use psilocybin because in the beginning, Charlie Grobe, who was a member of the Hefter board, Rick Doblin had recruited him to do a study of cancer patients with MDMA. And we met as the board with Charlie, and he said, you know, I'm not sure I want to treat dying patients with MDMA because it's a psychostimulant. It does increase your heart rate. It does increase your blood pressure. It has effects like methamphetamine to some extent. And he says, I don't know if I want to do that because these people are already in pretty dire circumstances. What do you think I could use? So we said, okay, well, what about LSD? And we said, if we use LSD, we're gonna have a media feeding frenzy. Because this is 1990, right? They're giving dying people LSD, this dangerous drug. So we thought the media would be on it like a fly on a cow pie, right? So we thought that's not going to work. Plus, it lasts all day long. So you're going to have to keep people in many cases overnight in the clinic, and they can't be discharged for another day. So that's going to add to the cost. Certainly, the early studies that were done with LSD, with say alcohol use disorder, it was shown that it actually had an effect there. But the duration of action and the publicity that we would get if we promoted a study with LSD was a problem. Now, I could have made the LSD because I was making it for my rat studies. We could have made it and made it available, but we thought, okay, let's not do that. Well, what about mescaline? The Native American Church uses mescaline, and from a peyote, they've used it. Mescaline lasts even longer than LSD, 8 to 10 hours. So again, and also a lot of people that take mescaline, they get very nauseous and sometimes even vomit. And people say that, you know, and the peyote cactus does it for sure, but synthetic mescaline will make a lot of people get nauseous and sick. So now we've got people who are dying. Do we want to give them a drug? And mescaline is not very potent. It's 200 to 300 milligrams, long acting, and makes a lot of people feel sick. So Charlie said, I don't want to make them feel sicker. DMT is not active orally. It's only active by injection, although it's very short acting. What about psilocybin? Well, that's been used for thousands of years in the form of magic mushrooms. Thousands of millions of people have used it. It's 4 to 6 hours. That's a much shorter frame than either LSD or mescaline. And nobody knew what it was. When we started doing this work, I was riding on an airplane with a psychiatrist. And I said, we were planning a study to give psilocybin to cancer patients. He said, "Psilocybin, what's that?" I said, "Well, did you ever hear of magic mushrooms?" He said, "Oh yeah, some people back in college were taking this. What's in magic mushrooms?" Nobody knew what psilocybin was when we started that. Now everybody knows what psilocybin is. Well, not everybody, but people on this call probably know. So it was really a question of practicality, some concept about how long people would have to be hospitalized, a little bit of concern about media attention and so forth. Now DMT, if you gave it by intravenous administration like Rick Strassman did, it doesn't last very long, and it's very powerful. People have these visions of alien beings that they can't identify. So I'm not sure that DMT would be very useful at least when we were starting those studies and the way we were planning to do it. Charlie gave psilocybin orally. All the studies with psilocybin have been done orally. So you get into a whole nother realm of complication. If it has to be given by intravenous infusion, then you have to make sure it's got sterility. You've got the whole thing about putting needles in and everything. So probably ayahuasca, which is really like an orally active psilocybin. If you ayahuasca, maybe, and I think it's probably 4 to 6 hours as well. So it could have been used, but in the beginning, when you're thinking about developing a medicine for FDA, if you think about what the FDA is looking for, purity, I haven't done any GMP synthesis, but I know it's a real pain in the butt. The MDMA and that I made for Doblin or the psilocybin, it only had to be super pure. I mean, we'd analyze it and get it, HPLC, and was 99.8% pure. But if you were given it intravenously, you've got another issue about sterility and who's going to do it, and people putting needles in their arms and so forth. So it was more complicated. With ayahuasca, you could probably do it, but ayahuasca is a mixture of things, and the FDA is going to want to know what's actually in there, how'd you make it, what are the individual components. We would have had to do a whole lot of analytical work, and the FDA, I'm not sure how they would have responded to a natural product mixture like that. I still haven't seen anything. Most of the work with ayahuasca has been done in Spain or South America, where they don't have the FDA here. So that's sort of, again, a long-winded explanation, but sort of how we ended up with psilocybin. But certainly psilocybin had a shorter action, and there are some psilocybin analogs that last maybe 2 hours, 3 hours. There are people looking at those. But shortening the effect, reducing the time that someone has to be in the clinic, reducing the number of sitters that have to be there, making it feasible for ordinary people to afford it. That's a big issue right now.
Thank you, David. Kevin White, you have a question. David, are you okay on time?
And what is it, 3? We can go for, I have my calendar marked off to 3:30. So
okay, great. Thank you. We have something from Rick, but it's shorter than usual. So if you're okay,
yeah,
we'll keep going. So Kevin White.
Hey, thanks. Firstly, Dr. Nichols, for your decades of really excellent work in the field, and we really appreciate your comprehensive knowledge of the science. The question I had sort of dovetailed with what you were just talking about, the purity of the MDMA that you had produced. There's been recent releases of information about how to formulate MDMA in novel ways by MAPS and other people. And I'm just wondering what your idea is about the capacity to achieve purity or potency levels using those techniques outside of a formal laboratory like your own.
I'm not sure what question you're actually asking. Are you asking about sort of the underground therapist getting pure MDMA, or?
Oh, MAPS and some other sources have released formulations for how to create MDMA in some novel forms right now. And I'm just wondering, what is the capacity outside of a formal lab where it's going to be regulated or illegal to do that? what is the capacity for a homegrown chemist or an official chemist to actually produce high quality MDMA using those formulations?
It wouldn't be that hard if they were a good chemist and they had integrity. If I think about what we did things in the lab that, except for the analytical determination at the end, that could have been done. The starting material we bought is no longer commercially available, but they can be made. So a chemist, and it could be made in a fairly large scale. A lot of underground chemists have equipment that'll accommodate 2, 3, 4, 5 liter flasks and things like that. And the chemistry is fairly simple, but the precursors are what are called watched chemicals. So you need to make this piperonel acetone to start with, which you can make, but you have to make it from methylene doxybenzaldehyde, which may not mean anything to you, but it's a watched chemical. So if you had a lab you set up in your basement, you wrote to some, and you get a letterhead and say, this is Kevin White, analytical consulting company, whatever, and you could write to Aldrich Chemical Company and say you want to buy 5 kilograms of piperonel, which is methylene doxybenzaldehyde. They would forward that on to the DEA. It's like, well, we've got this. He's got a letterhead and everything, but he wants to get a huge amount of this stuff. So then you'd get a knock on your door from the DEA, and they'd say, "Hey, Kevin, we see you ordered 5 kilograms of piperonel from Aldrich Chemical Company. We're just wondering what you're going to do with it." If they would send it to you, I'm not sure how this actually works as a watch chemical, but you could make that, and you'd only need hydrochloric acid and powdered iron and some nitroethane. So you could do that. And then you need it. Once you get that, you purify it and you make this piperonel acetone. Then you need methylamine. Methylamine is also a watch chemical because you can use it to make methamphetamine. So then you order a kilogram of methamphetamine hydrochloride, and now Aldrich tells the DEA, and they show up and they say, you bought 5 kilograms of piperonel and you just bought a kilogram of methylamine. We're concerned because these are the starting materials for making MDMA. Maybe you could enlighten us to what you're doing with this. So, but if that didn't happen, you could make it. And the way we made it was we condensed piperonel acetone with methylamine. You can do it with cut up pieces of ordinary kitchen aluminum foil. You put it in with methylamine hydrochloride and it reacts really nicely and you get a good yield. No, you work it up, have some big equipment. And then we vacuum distilled it. So if you were an organic chemist and you were making this stuff and you wanted to purify it, you could set up a vacuum distillation. You could buy equipment to do a small vacuum distillation. We were distilling, we made them 1.5 kilos. So we did 2 batches of it. You vacuum distill it and it looks kind of like a light colored honey. And then you just make it. All right. So you could actually do all those things. The problem is you'd run into precursors that are watched. And if, maybe you could smuggle in some from China, I don't know, or from Mexico, but you're really talking about setting up a kind of a criminal enterprise the way it's running now, and you'd probably get caught. You could do it, and it wouldn't be that difficult, but getting the equipment, setting up the lab and buying the precursors would probably raise some flags at the Drug Enforcement Administration. Is that the answer?
Yeah, the MAP formulation suggested that they were presenting compounds that weren't watched chemicals. There were novel approaches, but I think you've made the point that it's considerably more complicated than most people are going to be able to achieve.
And if you were a home chemist and you wanted to make 5 or 10 grams of it, you could probably get the precursors and nobody would pay attention because they're not looking for somebody buying 10 grams of piperonel. A lot of organic chemical labs would have it because it's used to make a lot of things. So if you were a graduate student in an organic chemistry department, you're walking through the storeroom, you could say, "Hey, there's some piperonel, there's some methylamine." You could probably make 5 or 10 grams for yourself and your friends, and you probably wouldn't get in trouble. It wouldn't be that hard. But if you're going to large scale, it'd be ringing some bells.
Okay, thank you, David. Rebecca Slater, would you like to ask your question?
I would. If you're going to larger scale, I highly recommend watching all seasons of Breaking Bad first, and grateful your name is Kevin White novel.
By the way, as a person who knows chemistry, the big problem with that, and he had a technical advisor, but you know, his methamphetamine was blue. And the only way that happens is if it's contaminated with copper. But they thought, oh, that'd be really cool if it's blue. That's how they would know it came from his lab. But actually, that would be a big mistake. It wasn't pure if it had blue color in it. So anyway, that's
Thank you for that. My family will enjoy that Breaking Bad piece of trivia. Seeing here first. And I appreciate you taking this question. So I'm interested in regarding of how different people suffer from trauma differently. You meet someone and they can manage and they can get about their day, and some people just have a hard time holding it together. I'm curious with the various compounds that are out there. Can you speak to how different compounds taken in a different order, maybe starting out with MDMA in one and moving into psilocybin, might change in the way that the trauma healing might unfold, if that does or does not play into it?
well, you know, with MDMA, it's very effective with therapy for PTSD. Usually it involves 3 therapy sessions, the way they've done it. In principle, maybe some others, maybe psilocybin would have some effect, and there are people looking at psilocybin for treating trauma. MDMA is kind of unique because what it does, in brain scanning studies years ago, they showed that the level of activity in the amygdala goes down after MDMA. The amygdala, if you electrically stimulate an animal, it produces intense rage and fear. In humans, it's thought that's where the fear comes from. So the idea with MDMA, I think, is the problem with trauma is if you go back and something is a trigger that caused you to relive the trauma, all the fear that's associated with that comes back and you're terrified. What happens with MDMA is it dampens the fear. And so what happens, in my understanding at least, is you can have a memory of it, but it's not associated with the affective fear component that came. So you can talk about it and understand it in that way without the fear. Psilocybin and LSD and these other drugs don't have the same effect on the amygdala. In fact, you can have a panic reaction with psilocybin or LSD, a fear reaction. So I suspect that with respect to trauma, MDMA is probably the best, although I know there are people looking at psilocybin for trying to treat PTSD, but I think that MDMA has a unique neurochemistry in terms of what it does in the amygdala, which is important for treating trauma.
Thank you for that explanation. I appreciate it.
One from Tim Buckley, which is a follow-up, I think, on the DMT question. Tim, do you want to ask your question?
Yeah, thanks. I put it in there before you you started talking about the IV questions, but so I don't want to belabor it, but would using IV psilocin, would you have enough control over the pharmacokinetics that you could kind of deal with this scalability question by shortening the trip significantly? And would producing IV psilocin be much more cost effective and thereby help keep the cost to the patient down?
Actually, I was the director of drug discovery for a biotech startup called Elucis Therapeutics, which did not survive, unfortunately. But the product that we developed that was an idea of mine was giving an infusion of psilocin, which is the active species. So we actually we had a GMP synthesis. We made psilocin benzoate, which is a salt. It's like you've heard of hydrochloride or whatever, benzoate. It had stability, it had solubility, and we had pharmacokinetics mapped out to show like how much you would give by infusion. It was much less, it was maybe 2 or 3 milligrams total, and it would hit this sweet spot where you presumably get the effect and then would drop off in a short period of time. Unfortunately, the company was sold to a company called Beckley Cytec, and their product is a 5-methoxy-DMT by nasal instillation. And I don't think they've done anything, but in theory, that was our idea, that we could give psilocin intravenously and we could control the infusion and that would shorten it. that hypothesis has not been tested, unfortunately. But yeah, your idea is a good one.
Thank you, Tim. Marcia Zuckerman, you have a question. Would you like to, you still okay, David?
Yeah, we still have 20 minutes.
Yeah, thank you.
Thank you very much. There do seem to be some people who don't respond to the serotonergic psychedelics and who may respond to other things like ketamine. Is there any understanding of that, and particularly whether long-term use of SSRI, although not concurrent use, might prevent a response from serotonergic psychedelics?
People who are on an SSRI do not have the same magnitude of response to psilocybin that someone who isn't on it does. They used to think it was incompatible, and now they've just basically shown, there was a study recently where they showed that people who had been given escitalopram, they had a somewhat blunted response, but it wasn't blocked completely. There may be people, and I've heard of people who are given big doses of psychedelics who had no effect at all and they weren't on any drug. Remember that we're all unique individuals, and the serotonin 2A receptor is a protein, a peptide, and you could have a mutation, what's called a single nucleotide mutation that would change one of the amino acids in that receptor, so it was no longer responsible to a psychedelic. So that's certainly possible. So, and there are people that have just didn't respond to psilocybin or even LSD, and people go, yeah, he didn't respond. We gave him a huge dose and nothing happened. So it's possible that in those situations, more than likely, there's a mutation in their serotonin 2A receptor, and that just doesn't respond. But generally, as far as I know, they've told people to quit taking psychoactive drugs before they give them psilocybin or LSD. But a study recently did show that people who are on escitalopram did not have a problem when they were given psilocybin.
Thank you. Dawn DeCuna has a question. You want to ask your question, Dawn? Dawn.
Where's Dawn?
Apologies. My question was really about dendritic growth, and I wondered if the growth is primarily in the pyramidal cells, or is it throughout the brain, or is there preferential growth in areas of the brain, and what are the implications of that functionally in terms of behavioral treatment or applied work, for example?
Most of the studies that have been done in mice, and they basically have looked at cortical cells, pyramidal cells. They are the major computational units. So that's what people have primarily looked at. They haven't gone in detail in other parts of the brain. I think they've looked at hippocampus, but primarily prefrontal cortex. And dendritic growth there, it's neuroplasticity is what they consider it. And there are things that can stimulate neuroplasticity. It's usually activation of brain-derived neurotropic factor, BNF, and that leads to activation of a receptor called TrkB, and that stimulates a neuronal regrowth. So I'm not aware of other areas of the brain because that's where people have mainly looked at, because that's where they've got the hints of activity in patients in postmortems, in patients who responded to SSRIs before and after, where they've seen an dendritic regrowth in the hippocampus. But I would assume the hippocampus, possibly the amygdala, the cortex, they all have pyramidal cells. Those are the major in the core, all throughout the cortex. Those are the major cells. But somatosensory motor cortex, I mean, they have pyramidal cells, but they do a different thing than in the prefrontal cortex, where they're more involved in cognition and executive function.
Thank you very much.
Thank you, David. And so Shippen has a question, so I'll move to the side and let him ask his question.
David, thank you so much for your presentation. I was recently at a gathering where Rick Doblin was presenting his research to a group of people, including a representative from a major medical school. And that person got quite excited and said, well, he might incorporate it in the curriculum. if they can figure out the mechanism of action. And my listening to you, I think there's probably a pretty good idea of the mechanism of action, but it leads to the broader question is, what is the current state of interest in medical schools and PhD psychology programs in incorporating the science into their curriculum? Can you give us some sense of what that looks like?
And I would add to that, if they're not doing it, how do we work on them to do it?
Yeah. Medicine is one of the most conservative fields. And psychiatry within medicine is the most conservative field of medicine.
Really?
Yeah. They're reluctant to try new things. They like to stick with what's out there. I don't have a very positive view of psychiatry. I hope there's no psychiatrist tied in.
I'm not a psychiatrist.
I know Duke University was trying to start a psychedelic research center. Duke is close by, in Durham, close by.
Mhm.
And he said that they've been stymied. There was actually a group set up, person who led it was trying to get approval. And the Duke attorneys said, no, we're not going to let you do that. They had arranged to get some psilocybin from an organization, a not-for-profit in Wisconsin called USONA Institute. USONA was going to supply it to them. And the legal people at Duke, which is a private university, said, they told USONA, you have to take responsibility if we have an adverse effect. USONA said, no, we don't do that. We'll supply you the psilocybin, but we're not going to take legal responsibility. You'll have to take it. So then the Duke attorneys said, no, we're not doing it because we're not going to accept the responsibility. There's another group at UNC Chapel Hill who I'm more closely connected with. And they've sent in a grant to try to get funding for a study. They have a lot of people interested there. I was down at the North Carolina Capitol a month or so ago, and they had a group down there that was trying to get an appropriation, and I work with the state legislator. They're trying to get an appropriation of $5 million, which would fund a study of psilocybin and a study of MDMA, and would provide some money for a administrative structure. Now that just got the first reading. It got through the first reading in some group, which I was there. There's two other groups within the state legislature that would have to approve it. And they're fairly negative in terms of whether they would approve it. But the fact that we could get this introduced in the state legislature in this initial committee, and that there was only one, we thought when the chairman of the committee said, okay, I read it, this is what it is. Any comments? There were two people in that committee that said, we support this. No one was opposed to it, which we were surprised. We had a bunch of people there ready to testify. I was there ready to testify,
right,
in terms of safety. So I don't know where that's going to go. UC San Francisco has actually got a psychedelic research center.
Mass General.
Mass General, yeah. We'll take that at Harvard.
Johns Hopkins has got a privately funded psychedelic research center.
Right.
So the things are happening a little bit slowly, but much more quickly than I ever imagined, actually.
Yeah, I think, David, my concern is that everybody's scrambling to train people if as and when these medicines become legal, but until you have the institutional infrastructure behind it, it's going to be a real uphill battle.
Oh, yeah. And MAPS has been, they've had a training program. I've also taught pharmacology to Mind Medicine Australia. They also have a training program. And also the California Institute of Integral Studies, CIS, they have a training program. And I've given lectures of pharmacology to all those groups. But the class is typically 100, 120, 150. And I think Doblin said they need 7,000 or 8,000 therapists. And so we don't have near the therapists. Now, we keep training people. Hopefully, there will be a place for them. MAPS is expecting to get approval by the FDA for MDMA and PTSD next year. There still won't be enough. And again, the issue with the PTSD, then 3 sessions are what they've been using. So three sessions of 4 to 5 or 6 hours a piece. It's going to be quite expensive. So scalability is still an issue.
Thank you. Yeah,
Mhm.
thank you.
See, the next CIS class is going to be 400 starting in September.
Oh, wow. Well, they've already talked to me about giving another lecture then, so
Good. We'll see you there.
you need 10 years of classes, 20 years of classes to get 8,000 therapists, right?
But we're growing quickly.
Yeah,
And there are lots of more training programs now too. So that's good.
yeah. No, it's, you know, it's a nascent field that everybody's excited about. And I think this is probably what happens with any new field. You know, a lot of false starts and back and forth. But I'm hopeful. A woman asked me 10 or 12 years ago, before all this came to be, and she says, what do you see as the future for these? Because I was working with psychedelics. Nobody's working with psychedelics. So she thought, like, why are you working on them? And I said, well, probably in the future, long after I'm dead, you'll be having a midlife crisis, and you'll go down to your primary care physician, and he'll refer you to a shaman / psychiatrist, and he'll give you a session with a psychedelic, and you'll get a new perspective on your life. She said, oh my God, you think you'll be dead by then? And I said, yeah, probably. But if the vector is pointing in the right direction, that'll be okay. Another woman asked me, why are you studying psychedelics? This has been like 15 years or so ago. And I said, think of the things that can change your life. You fall in love, you have a child, a sibling or child dies, parent dies, or you take a dose of LSD. Would stop, and it doesn't make sense. I said, how is it possible that a minuscule amount of a substance can go into your brain and change the way you perceive the world, either for better or for worse? I mean, how is that even possible? And I said, that's the thing that fascinated me. What is it in the brain? What's the importance of this receptor? Before we knew it was a serotonin 2A receptor. That to me is still an amazing challenge. And I'm surprised more people haven't gotten into it. Well, now they're starting to get into it, but for so many years, it's like, this was such a question. There wasn't, for academics, there was no funding. That was the basic issue. I was lucky to be in the right place at the right time. For a lot of academics that I knew, they wanted to do research in this field, but they said, there's no funding for this. So,
Right. Well,
What a trip.
I think that's a wonderful way to end. Knowing how powerful and important these substances are, and just huge gratitude, David, for all you've done in this field and for sharing this with us today. We really, really appreciate it.